process information, cells need the right proteins for the right job. In case study number 5, we ask the question, do all transcription factors bind to the same place in a promoter? If you remember from the previous case study, we found that in this particular promoter, all 4 proteins had to be present. But what we don't know is were all 4 proteins, the 3 transcription factors shown as the 3 colored circles, and RNA polymerase, did they all bind to the same place on the promoter, or perhaps the transcription factors bound to 3 separate places from the RNA polymerase? The investigators thought, well, they could design an experiment to test these 2 extreme possibilities. If the transcription factors bind separately to 3 separate locations in the promoter, then if you truncate or delete a part of the promoter, that sequence would be lost, the transcription factor could no longer bind there, and you would produce that the amount of transcript, the amount of mRNA would be reduced. If you deleted even more of the promoter, less of the transcription factor could bind, and therefore transcription would be reduced further. However, if all the transcription factors bound to the same place in the promoter, you could trim some of the promoter off, and the amount of transcript, the amount of mRNA produced, would be unchanged. They had 2 extreme ideas that they wanted to test experimentally. So they took a promoter as their positive control. It's 320 base pairs long upstream of the +1 nucleotide where the mRNA started. That's their positive control. And they measured the amount of transcript produced, but not by running a gel, but by looking at the phenotypes. So there were 2 phenotypes they considered. How fast did the cells divide and double their population, and were they resistant to a particular drug? So 2 different ways of measuring mRNA production levels by 2 different phenotypes. They got full transcription. When they deleted 165 base pairs of the promoter, they noticed no difference in either phenotype, therefore the mRNA level was unchanged. There's no transcription factors binding to this portion of the promoter. When they deleted 207 base pairs of the entire promoter, they got down to a promoter that's only 113 base pairs long, and this time they noticed that there was no drug resistance and the redoubling time had gotten longer. Therefore, the amount of mRNA had been reduced between the promoter that was 155 base pairs long and the promoter that's only 113 base pairs long. Therefore, they deduced in this region between -155 and -113, transcription factor or factors must have been bound to the promoter to regulate the amount of RNA polymerase bound and the amount of mRNA produced. They deleted 242 base pairs of the wild-type promoter. Now the promoter is only 78 base pairs long, and you notice there's no change from the previous promoter that was 113 base pairs long. Therefore, they deduced there were no transcription factors bound between base -113 and -78. In the final construct, they deleted 291 base pairs. Now the promoter is a tiny 29 base pairs. There's no growth at all, no drug resistance, so once again they had reduced the amount of mRNA produced. Their conclusion was that another transcription factor or group of transcription factors were binding to the DNA between bases -78 and -29. So for this particular promoter, there are 2 regions of the promoter sequence where transcription factors were bound. And remember, transcription factors bind to specific sequences. So this case study asked the question about transcription factors, and we learned that they bind to distinct sequences in a promoter. They don't have to bind all in one group. They can bind to different places scattered along the length of a promoter.